let's talk about what is enzyme affinity, and let's talk about how we can actually calculate enzyme affinity. When we talk about affinity, an easy way to understand affinity is the attraction of the enzyme for the substrate, right? That's a very vague definition. It's not a 100% accurate definition, but that's a general, simple concept of it. Now, to understand what I'm talking about, we first have to go over a definition. Enzymes have something that we call a turnover rate. What is a turnover rate? A turnover rate is, when you talk about it, a typical enzyme molecule can convert around, usually a normal enzyme, like a normal enzyme can convert around 1,000 substrate into product, right? It can convert around 1,000 substrate into product. That's the normal amount that we normally see. So turnover rate is the amount of substrate converted by enzyme per se. That's what turnover rate is. So how much substrate is being converted by enzyme per substrate? Average is around 1,000, but we have some enzymes that are working really, really fast, right? So when you go ahead and talk about it, now, how can we actually measure this, right? Or when we talk about how an enzyme is working or how fast an enzyme is working, what is the rate at which the enzyme is working, right? So how can we talk about, how can we discuss that, or how can we calculate the speed at which an enzyme is going to be working? How can you and I, when we take a look at the body, I told you around 1,000 substrate per second is common. But some enzymes, for example, can do 600,000 molecules. It's going to depend upon the enzyme itself. What is the rate of reaction for that enzyme? So how exactly are we going to go ahead and figure that out? Now, to figure that out, we first need to talk about Vmax. What is Vmax? Vmax is basically the theoretical maximum rate. The maximum rate at which an enzyme is going to be acting, that is your Vmax. And if you guys remember, in the last class, I had even drawn a graph for you guys where I had talked to you about, it was a graph like this. And I had mentioned on that graph that this is my graph. I have over here my rate of reaction. And over here, I have the concentration. And I had told you the shape of the graph would look something like this. And if you remember, I had mentioned that this particular point, this maximum rate reached by the enzyme, the maximum rate reached by the enzyme is going to be known as your Vmax. So that's what Vmax is. The theoretical maximum rate for an enzyme, the theoretically maximum speed at which the enzyme is going to be catalyzing reaction, right? Theoretic maximum rate at which an enzyme will catalyze the reaction, right? Now, if we go ahead and we talk about it, how can we use the concept of Vmax to calculate the rate of reaction? How can we go ahead and do that? Now, for that, we have to understand what Vmax is. Vmax is the maximum speed. Meaning that if I increase my substance anymore, if I complete the substrate concentration anymore, if I increase the substrate concentration anymore, my reaction will not get faster. And if you remember, I had told you guys that there's a reason. Why is it that even though I'm increasing the substrate, my reaction is not getting faster? What's the reason behind it? I had mentioned to you guys previously as well that the reason is because the enzymes active sites are all occupied. Because for the reaction to happen, the enzyme needs to have three active sites, right? If all the active sites are full, so it doesn't matter how much substrate I'm adding. It doesn't matter how much of the substrate I am putting in my reaction because the enzyme is fully occupied. The enzymes are fully busy. They are working at the maximum possible speed they can. They can't work any faster because they don't have any free enzymes. So Vmax is the point where all enzyme molecules, where all your enzyme molecules are bound to the substrate molecule. So all our enzymes are completely occupied. And now, if you wanted to measure this Vmax, because that's what I said, that one of the ways we can use to measure the rate of a reaction is to calculate Vmax. How can we calculate Vmax? Now, to calculate the Vmax, we will have to do a few steps. Step number one, we will have to calculate or measure the rate of reaction at different substrate concentration. So I will change the substrate concentration. Okay, I'll keep on increasing my substrate concentration. I will go ahead and I will take it, right? I'm going to go ahead and I'm going to take it from, let's say, 0 mole per dm cube. Then I increase it to 5 mole per dm cube, 10 mole per dm cube, 15 mole per dm cube. You will see that I am increasing the substrate and I'm increasing it by the same amount, plus 5. 0, 5, 10, 15. In the top, this is important for you guys to note small things like this for your paper 3 practical exam that you guys will be giving. And inshallah, later on, we will discuss as well how to solve your practical questions as well. But in practical exams, when you are designing your experiment, when you are doing your experiments, we have to make sure that we use a constant interval that's recommended. So we have to make sure that we use a constant interval. We are changing the substrate concentration by a certain amount, right? So in my case, I am changing it by 5. Now, I will change the substrate concentration and I will measure the initial rate of reaction. So at each substrate concentration at 0, what's the rate of reaction? At your 5, what's the rate of reaction? At 10, what's the rate of reaction? I'm going to go ahead and I'm going to do all of that. Now, after I go ahead and I do that, what next, right? What am I going to do next? How am I going to continue on after that? So, after that, we have to make sure that while we're doing this process, the enzyme concentration is constant. Okay, I can't change the concentration of the enzyme. The substrate concentration is the only thing that's going to change. This enzyme concentration is one of your control variables, right? So, and that is also important because when you guys are again doing your practical exam, when you're doing your practical questions, you have to make sure that only the independent variable is the variable that's changing. So in our case, the independent variable is the substrate concentration. Independent variable is the variable that you can change. Remember that. The substrate, that substance that you can change, the substance that's in your control, that's the independent variable. So substrate is in your control, you can change that. And the dependent variable is what you will be measuring. So we are measuring the rate of reaction. Anything else beside this is pretty much control variables. So enzyme concentration, I want to keep that constant. Temperature, I will want to keep that constant. pH, I will want to keep that constant, right? The reason for all of these things being is because all of these can affect the rate of reaction. So if you want your experiment to be a valid experiment, if you want your experiment to be comparable, you have to make sure that only this thing that you're investigating, so we are investigating the substrate concentration, so only the substrate concentration is changing, right? We have to make sure that the other factors are remaining constant. Only then, when you come up with your results, you can actually say that my result is about substrate concentration, right? If everything else is same, if anything else is different, if the temperature is different, then I can simply ask you, your temperature was not the same. So how do you know your result is not because of temperature? How can you be sure it's because of substrate, right? So we want to make sure that in paper 3 questions, not just in paper 3 questions, in future, later on, when you guys decide to pursue your, post-graduation, graduation, post-graduation, whatever, whenever we do any experiments at all, any research work, we always want to make sure that all of the variables are the same, right? So enzyme concentration, temperature is constant, pH is constant, fine. We are going to go ahead and do that. After, what is going to happen, that as the substrate concentration is going to increase, so your rate of reaction is going to increase until it reaches the maximum speed. That's common sense. As we increase the substrate, the rate of reaction is going to increase until it reaches the maximum velocity. So after this, what are we going to do? We are going to plot a graph, right? Plot a graph of initial rate against substrate concentration. So you will plot a graph, initial rate of reaction is on the y-axis, the substrate concentration is on the x-axis. And as a result, you're going to end up getting the curve that I've drawn. Now, you guys would say, okay, miss, so that's it. We plot the graph, and after plotting the graph, we end up, we can just read from the graph what the value is. In theory, it seems simple. In practice, there is a small problem. In practice, the problem is that the curve doesn't actually look like this, right? The curve looks more like this. Let me just draw it. Hold on. The curve looks more like this. So it technically, theoretically, it should reach a stage where it stops increasing. But in practice, it sort of becomes flat, but it still continues to rise. It doesn't become completely horizontal. It will become horizontal, it will become flatter and smaller and smaller and smaller, but it just doesn't become 100% flat. Gradient doesn't end up becoming zero. It still continues to increase by a small amount, right? This is, so in mathematics, you don't have to remember this, but just letting you know, in mathematics, a graph like this is basically known as an asymptotic curve, right? We have a mathematical term for it. It's an asymptotic curve. Now, when you one of the properties of such a curve is that it never actually flattens out. So that's what you're going to observe. In reality, theoretically, this is what you and I should see. But in reality, this is what we would see, that it actually doesn't flatten out completely. It will flatten out quite an extent, but it still continues to increase a little bit. So I can't just read the value from the graph and be like, this is the Vmax, right? That's the problem. So what is the solution? how can I salute it? How can I figure out my solution? The solution to my problem is actually quite simple. The solution is I can plot a graph, but not a graph of rate of reaction and concentration. I plot a graph of, I go ahead and I plot a graph of 1 upon S. So 1 upon substrate. I take the same substrate concentration, but I do 1 upon S. And I plot a graph of 1 upon R. So I take out the reciprocal, sorry, I said R, I meant velocity, because we're measuring the speed of the reaction, right? So I can go ahead and I can take my 1 upon V and I can take 1 upon S. So this type of a graph is known as a, or the better option is plot a graph of 1 upon V against 1 upon S. And this type of a graph is known as the double reciprocal, because I'm taking two reciprocals, right? Both the x-axis and the y-axis are a reciprocal. So this type of graph is known as a double reciprocal graph. So I can plot this. When I will plot a double reciprocal graph is that, so what's the advantage? Is that so the problem with this graph is that I can increase the substrate concentration to infinity, because my graph will never flatten out. I told you that's the mathematical property of this particular graph. It will never truly 100% become horizontal. So I can, if I want to continue plotting it, I can continue plotting it to an infinite value, because it's never going to become horizontal, right? Now, what is going to happen is that when I'm going to plot a graph like this, I will end up getting a straight line graph. That's the advantage. So instead of having to bother myself with a curve, I'm going to end up with a straight line graph. And you guys will see exactly how that is important for us. We will end up with a straight line graph that is going to be looking something like this. So it's not going to go through 0, 0, by the way. It's going to go like this. This is the graph that you're going to end up with. 1 upon S and 1 upon V. Now it's of course much easier for you to read the V max, right? How are we going to read the V max? To read the V max, you would think that you need to read the maximum velocity, but you need to realize that we are doing reciprocal. So our V max is actually this, my y-intercept. The y-intercept given to me in the graph, this y-intercept is going to be my V max. So if I go ahead and I make this double reciprocal graph, I can then do this. So all I have to do is my measurements and my experiment remains the same. But when I'm plotting the graph, I will just take out the reciprocal of the values. That's it. And I'll plot my graph, right? So this is V max. This particular point is going to be V max. Okay. This is what you guys can just learn. You guys can just learn and say, okay, this is the point where V max is, and that's it. If you guys don't understand the theory behind it, that's okay. It's not going to affect your past paper questions. But let's just for the sake of explanation discuss why. Why is V max over here and why not over here? So theoretically speaking, I told you guys that the graph of the curve of your rate and concentration never flattens out, right? That's what I told you guys. That's the problem with it. It never flattens out. So if I'm actually doing the experiment in real, I can keep on increasing the substrate concentration till I get to an infinite value. Now, so that is something that's obviously you can't do. You can't just keep on increasing substrate until you are with an infinite amount of substrate, right? That makes no sense. How are we going to do something like that? But I want to know what is the rate. or what is the velocity at infinite substrate, right? That's our goal, because our curve is never going to become flat. It's going to keep on increasing as I increase the substrate. It's going to be increasing by very small value, but it's still going to be increasing. Really minor value, minuscule value, but it's still going to be increasing. So now that means we need to find the velocity or the rate at infinite substrate concentration. If you guys remember a little bit about the mathematics that you have done before, you might have studied that if we divide any number by 0, we get the value of infinity. And if we divide any number by infinity, we get the value of 0. So I'm doing 1 upon s. So I want to find out what is the maximum velocity at infinity. When I have infinite amount of substrate, what's the Vmax? Right? That's what we have to calculate. So if I do 1 upon s is equals to 1 upon infinity, I get 0. So I will be looking the value over here where the 1 upon s is equals to 0. So that's how the reciprocal graph solves the problem. That's how by taking a reciprocal, my problem is solved. Because if I'm doing 1 divided by infinity, that is equals to 0. So if I make a graph of 1 upon v and 1 upon s, I simply need to look at 0 to calculate where the Vmax is. That's the theory behind it, but if you find that the theory is too confusing, the actual, you can just remember that this point, the y-intercept, when your 1 upon s is 0, that is the point at which you can now obtain your Vmax. So your y-intercept is going to be acting as your Vmax, and we can write that down. Your y-intercept is the So that is one possible way or one thing that you guys can do to calculate the rate of the reaction. Now, when I talk about this graph, the story has not ended as of yet. There is more to the story. How can there be more to the story? The way there is more to the story is like so. One of the information I can get from this graph is, I just told you, we can calculate the Vmax, right? From this exact same graph, I can calculate another interesting piece of information. What is the other interesting piece of information I can calculate from this graph? So the other interesting piece of information I can calculate is a constant. It's known as the Michaelis-Menten constant. Let's go ahead and talk about it. What is this particular constant and how does this help us in our scenario, in our calculations, in our topic of enzyme reactions? How is this helping us? How is talking about the Michaelis-Menten constant helping us out? Let's take a look. So, Michaelis-Menten. I hope everything is making sense so far. Anuj, Khadija, is this all clear? How we're solving the question? Sorry, how we're the topic, like it's making sense so far? How we're doing the what we
Yes,
saw?
yes.
Okay, all clear. Okay. So the next piece of information that I can get from this graph is I can find a particular constant that is Michaelis-Menten constant, and the symbol for this is Km. Okay. Now, what actually is the Michaelis-Menten constant? Michaelis-Menten constant is the substrate concentration, the substrate concentration at which an enzyme works at half its maximum rate. So the maximum rate that you have, if you half it. So what is the substrate concentration I need for that? What is the substrate concentration at which my enzyme is going to be working at half the speed? What is the substrate concentration at which my enzyme is going to be working half its maximum rate, right? So what actually is this? This is the point at which half of the active sites are occupied. So I want to calculate half of Vmax. Whatever the Vmax is, I want to calculate the half of it. That's basically what I want to know. And if I calculate half Vmax, that will tell me what the Michaelis Menten constant is for this particular enzyme. This is the point at which half of the active site are occupied. So at that particular point at which half the active sites are now occupied, that is the Michaelis-Menten constant. Now, why do we need to know about this? I mean, we talked about Vmax and we said that the Vmax is the maximum velocity or the maximum rate of reaction. Since we already have the Vmax, why should we bother ourselves with something like this? Why? What does it matter how the enzyme is working at half Vmax, right? I mean, we know the maximum rate. That's good enough. That's because this gives us a different piece of information, or we can interpret it in different ways. So, what is going to happen? Think of it this way. The higher the affinity of the enzyme for the substrate, right? The higher the affinity of the enzyme. So I told you affinity is sort of the attraction, how much that enzyme is attracted towards the substrate, right? So higher the affinity of the enzyme for the substrate, the lower the substrate concentration needed to reach half Siemens, right? So if we go ahead and talk about it, the higher the affinity, so the more an enzyme is going to have an attraction, the more that enzyme is going to be attracted for the substrate, so the binding between the enzyme and the substrate will be more, right? Because the enzyme is really attracted to the substrate. So there's going to be more binding between the enzyme and the substrate. So at less substrate concentration, you will reach half Vmax. At less substrate concentration, you will reach half Vmax as compared to what? As compared to your um, as compared to your an enzyme with lower affinity. An enzyme with lower affinity has less attraction. So to reach that same reaction, to reach that same half Vmax, we will need more substrate. For an enzyme with high affinity, I don't need that much substrate because the enzyme and the substrate are always, they are attracted to each other. So what are we going to see? We are going to see that we can use Km to calculate or see enzyme affinity. So Km actually tells us about the affinity of the enzyme. Vmax tells us about the turnover rate or the rate of reaction. Km tells us about the enzyme affinity. What is the affinity of this enzyme for this particular substrate? And remember this, higher the affinity, the lower your Km. Because like I told you guys, higher the affinity, the less substrate concentration is required to reach half Vmax, because the enzyme has a strong attraction towards the substrate, right? So what's going to happen? Higher the affinity, the lower your Michaelis-Menten constant and quicker your reaction. Faster my reaction is going to be, right? Now, keep this in mind, the maximum rate is not affected by Km. Keep that in mind. The Michaelis-Menten constant will not affect the maximum rate. The maximum rate will be the same. So maybe you have two enzymes. One has less affinity and one has higher affinity. The enzyme with low affinity will have higher value of Km. It's an opposite relationship. It will have a higher value of Km because that enzyme has weak attraction. Since it has less attraction, so you need more substrate. It needs more substrate to bind to the reach the value of half Vmax. Then you have the enzyme with a higher affinity. The enzyme with the higher affinity, you will need less substrate. So you will have a lower value of Km. Now, it is very much possible that the enzyme with low affinity, it will have a high Km. So you are need more substrate to reach the half Vmax. But that that particular enzyme could have, for example, a high turnover, a high Vmax. The maximum velocity of that enzyme, it has low affinity, but the maximum velocity of that enzyme maybe will be higher. The high affinity one can have a lesser maximum rate. So maximum rate is basically the maximum speed at which an enzyme is going to be working. Michaelis-Menten constant is about how quickly it will reach the maximum rate. So the maximum rate is not affected by Km. Km is not about maximum rate. So don't think of it this way, that if an if a enzyme has higher affinity, low Km, so since they have higher affinity, they must have a higher rate of reaction. No, that's not true. Not necessarily. Okay? The maximum rate, the Vmax, is not affected by the Michaelis-Menten constant. Okay, Michaelis-Menten constant simply tells me how quick the reaction will be. Vmax tells me what is the maximum rate or the maximum speed it's going to reach. Right, so there's a difference between the two of them. And so Vmax and Km are going to provide me with two completely different ways of comparing the efficiency of enzyme. So Vmax and my Km allow me to compare differently, they're not linked with each other, differently the efficiency of an enzyme. The Vmax is telling me what is the maximum rate at which a reaction is possible, what is the maximum, how fast, what is the maximum speed a reaction can occur at, while Km is telling me the affinity. What is the affinity for the enzyme for the substrate, right? Higher the affinity, remember this. Higher the affinity, the more likely that the product will be formed when a substrate molecule enters the active site, right? So the more, that's what affinity is. I told you it's about the attraction. The more likely that the product is going to be made when a substrate molecule enters that active site. In low affinity cases, maybe the substrate will simply leave the active site before the reaction takes place. Before the reaction has even happened, the substrate has moved away from the active site, right? That would be low affinity. High affinity is that when the substrate binds to the active site, it will turn into a product. So, we can use both. We can use both of these concepts to measure the efficiency of an enzyme. How efficient is it? First of all, how quick is it going to be working? And secondly, what's the maximum speed at which we can get it, right? A good way to understand this is if you think about a car. So a car, for example, one thing is acceleration, right? How fast the speed of the car is increasing. That's one concept. So how fast the speed is increasing, that is acceleration, that is Km. So Km tells me how fast my reaction is increasing, how fast my speed is increasing. Okay? And Vmax is the maximum speed of the car. So Km is the acceleration and Vmax is the maximum speed of the car. Think of it that way. So Km is going to be how quickly my speed of the reaction is increasing. So when I'm increasing my substrate from 1 to 2 to 3 to 4 to 5, how quickly the speed is increasing. That is Km. And what is the maximum possible speed I can get it? That is Vmax. And they're not going to affect each other, right? For example, the acceleration. It doesn't matter what is the value of acceleration. The maximum speed that I want to reach is the same, right? If I want to take the car to 10 meters per second, the acceleration will simply speed up the car. The constant speed or the maximum speed is still 10. So the same concept can be applied to enzyme. Now, you guys can go ahead and ask me the question, okay, miss, this is all fine and well, but you still haven't told us how to actually calculate the Michaelis-Menten constant. We have discussed about where it is, we have talked about what it is, but what actually where actually do I need to calculate the Michaelis-Menten constant? Where actually can I calculate the Michaelis-Menten constant? So remember this, the y-intercept is where you get your Vmax. And the x-intercept over here, this is your Michaelis-Menten constant. So this is where you're going to find your Michaelis-Menten constant. This is equals to, this point is equals to minus 1 upon km, the y-intercept, right? Your y-intercept is equals to minus 1 upon km, minus 1 upon km. Of course, this will also be in reciprocal because I'm calculating reciprocals, right? Because the entire graph is in reciprocal. So this will also be in reciprocal, but no problem. Once we know the value for minus 1 upon km, we can calculate the km very easily. It's simply a matter of rearranging the equation. So this is going to give me minus 1 upon km. And over here, I'm not going to get Vmax directly. I'm going to get 1 upon Vmax, right? And then once we have the 1 upon Vmax, then it's no problem. We can simply rearrange the equation and find the value for Vmax. So this point is 1 upon Vmax and this point is minus 1 upon km. What about over here? In this particular graph, what would you see? So in a graph like this, your Vmax would be a bit higher because technically the Vmax, like I said, in this type of a graph, the curve never truly flattens. So the Vmax is definitely going to be higher than where it seems to be. But what about Km? So Km is going to be when you are at half the substrate concentration. So if I talk about half Vmax, so half Vmax, for example, is let's say over here. Let's say this was 10 and this is 5. So at half Vmax, what is the substrate concentration I need? This is my Km. So this is the Km in a curve. If I make a curve, this is where I will get my Km. And if I talk about my reciprocal graph, double reciprocal graph, so the x-intercept is the minus 1 upon Km and y-intercept is the 1 upon V max, right? So that is what you're going to go ahead and actually see in your graph. Now, keep this in mind that Km is not a fixed value. What do I mean? For example, I have told you guys that we have this enzyme. Now you would say, after a while, I say that the value for this enzyme is changing. Yesterday, the Km for this enzyme was 10, minus 10, for example, or 10, whatever. Today it is 20. Now you will say, ma'am, the enzyme is the same. It's the same enzyme. How has the Km changed? How has this Michaelis-Menten constant changed? How has the affinity changed all of a sudden? You're using the same enzyme as yesterday. Why is it changing? Remember this. The value of Km for a particular enzyme can vary depending upon a few factors, right? What does it depend upon? What factors? So some of the factors that we can talk about is substrate concentration. Right? substrate concentration. Then we can have temperature, right? Optimum temperature, we will see the highest value of Km. Maybe the next day the temperature is now much low, so the affinity is not that high anymore. pH, right? Presence of some particular ions. Some ions actually affect the activity of enzyme, right? The presence of inhibitors. Do we have any inhibitors today? Do we have any pollutants? Do we have any poisons, right? So depending on all of these things, the value of Km can change. Km is not specific to an enzyme. Just because in the question you have an enzyme, a particular enzyme with a particular value of Kmax, sorry, your Km, so that can change. It's not fixed, right? The value for Km can actually change depending upon the situation. Now, that is what we have to do. Now, why did we study all of this entire situation? I told you guys it's to study the efficiency of the enzyme. But how is the entire efficiency helping out you and me? Why do we want to study the efficiency of the enzyme? There are lots of different things you guys can do if you decide to study about the efficiency of enzyme. There's lots of different things that you guys can do. For example, let's talk about it. Scientists can use this entire Vmax and Km calculations to actually make models on the computer about how a reaction will happen, right? So they can predict by using these values, they can actually predict, okay, this is how an enzyme is going to act. This is how an enzyme is going to be working. This is how the reaction is going to go if we add this enzyme. And that allows them, so we have a lot of enzymes that we even use in medical treatment. So they can study what is the optimum temperature, what is the optimum pH. They can study about inhibitors. If you remember, I told you inhibitors are used a lot in the medical field, right? In the medical field, I can use inhibitors to go ahead and do that. I can use inhibitors to help me with diseases. So I can study all of that by studying about Vmax and Km and all of these things. We can also study about the preference of an enzyme. For example, I told you in competitive inhibition, we have competition. Now you guys can see with the concept of affinity that if an enzyme has higher affinity for one substrate, so it will bind to that substrate, right? Competitive inhibition. So we can understand that. We can use it in genetic engineering. You know, in genetic engineering, we often need catalyst. So what is an enzyme? It's a biological catalyst. So we need it for that. We have a lot of commercially available enzymes. Enzymes that I told you we use in medicines, or enzymes that we also use in a lot of industrial processes, right? So in a lot of industrial processes like fermentation and all that, we sometimes need catalyst. So those catalysts, we can study those catalysts and by studying about Vmax and Km. So this, we can basically use all of these to then, so these are the different things that you can do by studying the entire concept of enzyme affinity, Km and Vmax. All clear so far? Anything that anybody would like to ask?
No.
Aruj, all clear? I hope no confusion. Okay. Now, the last thing that we are left with in this topic is that we have to talk about enzyme immobilization, and that's the last thing we need to do in this particular topic. So let's go ahead and just quickly go over this so we can finish off our topic of enzyme.
Miss, could you show the previous slide?
Yeah, sure. There you go.
Okay, let's start.
Okay, so when we talk about enzyme immobilization, why do we want to do enzyme immobilization? I just told you that we use enzymes in medicines. For example, if a person is not able to eat lactose products, then one of the opportunities that we can offer is that we can give them the enzyme lactase. So people who are naturally lactose intolerant, people who naturally cannot digest lactose, we can give them that enzyme lactase. They can eat it, and that enzyme can break down the lactose for them. Or we need it in food technology, or we need it for industry. So we need enzymes for different things. How actually can I get enzymes, and how actually can I get you know get my hands on these enzymes? So to do that, I will have to immobilize the enzyme, right? Now, when we talk about enzyme immobilization, so one of the ways in which you can immobilize the enzyme is, I just told you guys that one of the common things that we can use for is lactose intolerance, right? People who are lactose intolerant, we can give them the enzyme, lactase. So that lactase will help them in breaking down lactose. So now let's just take that for an example. How can I immobilize lactase, right? So the way that we can go ahead and do this is so that I generally will take my lactase enzyme, right? I will take my lactase enzyme and I will mix it with a compound that is known as sodium alginate. Okay? We'll mix it with a compound known as sodium alginate. Then I will take a beaker with a solution of calcium chloride. The experiment kind of looks like this. So this is my beaker, and over here I would have a syringe. I can remove the needle of it. Now, this syringe is going to be filled with a mixture of my enzyme and a mixture of my sodium alginate, right? And over here, I have what? I have my beaker. The beaker has a solution of calcium chloride. Now, what are we going to do? We're going to lightly press on the syringe, and we're going to let the drops of this mixture fall into the fall into the calcium chloride solution. What will that do? So what that will do is this, that your calcium chloride, the sodium alginate and your calcium chloride will react together to make a jelly-like substance, right? Will react together to make a jelly-like substance. Hence, each drop will turn into a small bead. Each drop will sort of harden and become into a bead or a jelly or a jelly sphere, right? Each sphere is going to harden and it's going to turn into a sphere. At this particular point, now our enzyme is immobilized. So it's not that hard, something that's actually quite easy to do. It's not that hard, right? Then what can we do? We can take these enzymes and okay, so then we can further use these enzymes for different things. I can either take these enzymes and I can put them in a tablet, right? I can put them in a tablet and I can give it to a person who would want to eat that enzyme. Or another thing I can do is that I can take these beads and I can pack them into a column, like you have a fractionating column in chemistry, something like that. So I can pack all of these inside a column. And From the top, I can add some milk. What will happen? What's going to happen is this, that at the bottom, I have my beaker. And as the milk is going to pour over the enzyme, all the lactose inside the milk will be digested. And at the end, I will get lactose-free enzyme. Sorry, lactose-free milk. Right? This is another application. So I'm just giving you an example. We can do a lot of things with this concept. I can do a lot of things with this concept. This is just one example. So this is one of the commonest methods that we use for immobilizing enzymes. And this is one of the commonest things we can do with it. That's done a lot in the industry, that we can use it to make lactose-free milk. So what are the advantages for, let's say, all of this? Why would I want to do enzyme mobilization? What are the advantages for this process? The advantages is that, so, okay. So if, for example, you can ask me, Miss, why can't I just take the lactase and why can't I just take the lactase and mix it with the mix it with my product over here, right? You can ask me that. Why is it that I need to do what you just said? Why is it that I need to go through the process of enzyme mobilization? Why can't I just directly mix the enzyme? Why can't I mix lactase with the milk? I'll get the same result. Why do you want me to go through this long process? So, compared to just mixing the enzyme with the substrate, enzyme immobilization basically allows or saves you from the hectic process of separating the enzyme from the product. So if I just mix the enzyme with the milk, after that, how will I separate out the enzyme? I will have to give the milk as it is to the poor people. And there's no harm in that, except for the fact that now I need to buy more enzymes. And my business will become really costly. I will end up going broke, right? Because I have to buy, each time I have to buy more and more enzymes. So this helps us in saving the cost of the product. My cost of the product is going to be saved because I'm simply mixing I don't have to worry about the enzyme mixed with the substrate. I can immobilize the enzyme and use the enzyme again and again and again and again and again. So it will save me cost, and also there will be no contamination, right? My milk is not going to be contaminated with the enzyme, right? Secondly, like I said, it is cost effective, right? These are the two things: no contamination, and it is going to be cost effective, right? I can reuse the enzymes again and again. Along with this, what other possible advantages can we have to enzyme immobilization? That the immobilized enzymes are more tolerant to temperature changes and pH changes, right? Remember this. Immobilized enzymes are more tolerant to temperature and pH changes, right? As compared to an enzyme in a solution. So if I take an enzyme and just mix it with milk, it's still going to be affected by the temperature and pH. Immobilized enzymes are also affected, but not that much, right? Why does this happen? Because the enzyme molecules are firmly held in shape by the alginate. So they don't denature easily, right? So that is the advantages that you have to do related to your topic of enzymes. And that is basically it. We are done with the topic of enzymes. So in this topic, you need to know about your in this topic, you need to have a good idea about your enzymes, in the sense that you need to know about the factors affecting the enzyme and the theory behind it in relation to protein structure. You need to have a very good idea about your inhibitors. Then you need to see a lot of questions on Vmax and Michaelis-Menten constant, and then this immobilization of enzymes. So these are the important topics that we had to go over. So we're done with the topic of enzymes. Any confusion? Anybody would like to ask anything? Khadija, Anuj, is everything all clear?
Yes.
Yes, miss.
Okay, guys, in that case, we're going to go ahead and stop it over for today.